This lecture, we are going to look at proteins in detail. I already sent a lecture on proteins before now. So we are going to look at the other material that was sent to you on protein. And in this lecture, we are going to go in detail into exploring what proteins is actually about. Now, I already sent a lecture on protein, but I just thought it wise to use the current lecturer's material to explain what protein is. Now, we'll talk about proteins. We say they are linear biopolymer. Now, the word bio, linear, of course, you know what linear is, straight chain. Bio, biological, polymer. Are you getting it now? I believe those of you in chemistry, you'll be taught polymer chemistry during your stay in the Department of Chemistry. When we say something is a polymer, that means it is made up of so many repeating units, which are called monomers. Anything that's a polymer is made up of so many repeating units called what? Monomer. As name imply, polyethylene. Polyethylene is made up of many ethene. That is where your polyethylene bag came from. So when we talk about biopolymer, they are biological molecules that are made up of several repeating units called monomer. And the repeating units that proteins are made up of are the L-alpha amino acid. Now, what do we understand by the L-alpha amino acid? Now, if you learned about the stereochemistry of amino acids in your first semester, you agree with me that you were told that most of the amino acids that are found in our body and in nature, that is the 20 amino acids, are alpha amino acid. Aside from proline. Proline is not an alpha amino acid. Now we say something is alpha amino acid. What does that mean? Of course, you already have an idea of it before now. If you have something like this, C, and then you have COOH. Most of the amino acids you have are in this format, COOH. And then you have the H2N, which is the amine group. And then you have the hydrogen. And then you have the R. Now, you learned in your first semester that this R of this amino acid is called the side chain. Are you getting it? The R is called the side chain. Now, why do we call it alpha? The predominant functional group here is the carboxylic acid functional group. So if we want to number, we number from here. So here will be number 1, here will be number 2. However, in chemistry, there is this common way of numbering, where we don't use 1 or 2, but rather we use alpha and beta. Now, if this is the predominant functional group, it means that any other carbon close to the predominant functional group is an alpha carbon. So this is an alpha carbon, and if this side chain carries a carbon, this other carbon here will be the beta carbon, and then we get gamma and so on and so forth. So this carbon here is the alpha carbon. Now, you agree with me that the alpha carbon has the amine group and the carboxylic acid group attached to it. So whenever you have the amine group and the carboxylic acid group attached to the alpha carbon, we say that such an amino acid is an alpha amino acid. L talks about the enantiomeric form of the amino acid. We have two enantiomers. We have the D enantiomers, and then we have the what? L enantiomers. We said enantiomers are non-superimposable mirror images of each other. Stereochemically, you'll be able to distinguish between the D enantiomer and the what? L enantiomer. But in nature, most of the amino acids that are found are usually in the L form. Is that okay? So the proteins, they are made up of the L-alpha amino acids. So when you have several L-alpha amino acids joined together, what you are going to form are proteins. And the bond that links them together is what we call the peptide bond. Is that okay? The bond is called the what? The peptide bond. You should take note of this. It's very, very important. We are going to dwell more on this later. Then amino acids that form proteins are 20. Like I told you that there are 20 amino acids that form protein. However, these 20 amino acids, they repeat themselves over and over and over and over and over again. Is that okay? They repeat themselves over and over and over and over again. That is the reason why you now see that we have several proteins with different lengths of amino acid. That does not mean that the highest length of amino proteins we can have is 20. We are just saying that the amino acids that form proteins in biological molecules or in biology, in biological systems are 20. Now, these 20, you can have repetition in such a way that you can have up to thousands of units of amino acids. Is that okay? Now, there's what is called the central dogma of molecular biology. When we talk about the central dogma of molecular biology, we are talking about how genetic information, which are the DNA, are converted to RNA, which are later now converted to what? To proteins. Is that okay? This is the process for manufacturing of proteins in the body. Proteins are manufactured from the gene, which is the DNA, which converts such protein to what? RNA to what? To protein. Now you are going to learn that later when you talk about DNA replication, then transcription, then translation, or DNA replication, translation, and then transcription. All right, this actually talks about how proteins are manufactured in the body from the gene. This is what we know as the central dogma of molecular biology. And of course, you know that proteins are major component of the cells. Proteins are major component of the cell. I have explained that in our first lectures, which I sent earlier, that the cell membrane is specifically made up of proteins and lipids and a little amount of carbohydrates. However, by mass, the amount of proteins we have are much more than the lipids and the carbohydrates that are present in there. Is that okay? Some of all these other organelles, they are made up of what? Proteins. So that is that. Now, the next thing we are going to look at now is what actually forms protein. Now, we said proteins are made up of what? Amino acid. Now, when these amino acids join together, they are called a peptide. Now, several of these amino acids joined together are called polypeptide. Now, proteins are made up of polypeptides, which can be up to 100 amino acids repeating themselves to give us a polypeptide, or more than 1,000 amino acids repeating themselves to also give us what? Polypeptide. So the polypeptide chain can be it can be 50, like that of insulin, which is about 51 or 50, and then we have other ones that are up to 100. Then we have the ones that are up to thousands. Is that okay? Those ones, they are called polypeptides. So we said proteins consist of one or more linear polymers called polypeptide. Now, a protein can be just one polypeptide. When we talk about the polypeptide, if this is an amino acid, you have another amino acid linked together, another amino acid linked together like this, up to 100 to 1,000 times. You say this is one polypeptide. So amino acid could be just this one polypeptide. Proteins can be just this one polypeptide, and proteins can be two polypeptide chain, often joined together by a disulfide bond. All right, often joined together by a disulfide bond. So this is one polypeptide. This is another polypeptide. So a protein could be just one of these polypeptides, or a protein could be a combination of this polypeptide and this polypeptide, which we have two polypeptides. Is that okay? It could be 3 polypeptides, it could be 4 polypeptides, but proteins are linear and are never branched. Proteins are linear and are never branched, and they are joined together by peptide bond. Now, what is the peptide bond? Let us take hypothetical amino acid. Now, when I talk about hypothetical amino acid, I just mean just any amino acid without taking into cognizance the name of such an amino acid. So you have this, and of course I can say this arrow group is the side chain. So if I have this and then I have another amino acid, my dear student, is COOH, and then I have another amino acid, this H, this COOH, R. These are three different amino acids. Now, my dear student, the amino acid, they have what we call the N terminal, and they have what we call, in every protein rather, we have what we call the N terminal of the amino acid, and we have what we call the carboxylic acid terminal. Now, the reason why we call them the N terminal and the carboxylic, or amino terminal and the carboxylic acid terminal, is because those ones, they do not undergo condensation reaction. For instance, this hydrogen, this amine group here, it does not bond with anything, so you can say this is what the N terminal. Then this carboxylic acid here too does not bond with anything, so this is the carboxylic acid terminal, which we can call the C terminal. Now, what actually happens in the formation of the peptide bond? Now listen, we have this water molecule will be given off. All right, water molecule will be what? Will be given off. So if water molecule is given off, my dear student, it means if we take, let's say, OH from here and take H from here, what are we now going to form? That is water. So I'm going to erase OH from here and then erase H from here. So if I elongate this, you are going to form a new bond entirely. So you have, what did I remove from here? OH from here. So we are having CO, and then we elongate this side, and then you have your what? NH or HN. This bond here is what we call the peptide bond. This bond here is what we call the what, my dear students? Is what we call the peptide bond. Peptide bond. Is that okay? This is what we call the peptide bond. Do you understand the concept now? Now, each of these amino acids that form the peptide bond, they are no longer called, immediately they form amino acid, they are no longer called amino acid. Immediately they form the peptide bond, rather, they are no longer called amino acid, but now you are now going to call them amino acid residues or peptide. Like this one now, these two, this you see here, we are no longer going to call them amino acid because they are no longer amino acid. They formed a peptide bond. Are you getting it? They've lost water via a condensation reaction. Via what kind of reaction, my dear students? A condensation reaction. You are no longer going to call this an amino acid, but you are now going to call it a residue. That is what is left after the peptide bond is formed. So you call them amino acid residue. Is that okay? Amino acid residue. Is that you call them amino acid residue, my dear student? Or you can call them peptide. Or you can call them what? Peptide. So in this case, this the one you have in your material, if you count very well, go to page 2, you will see one peptide, another peptide, another peptide, another peptide, and another peptide. There are 3 peptides there. So you call that a what? A pentapeptide. Is that okay? Pentapeptide. Now for this one is just a dipeptide, because this is one peptide. This one, you can say this is one. This group here, this one here, are you getting it? Is one peptide. This is another peptide. You see that 2 peptides are joined by peptide bond. The bond is called the peptide bond. Why the residues left, they are called what? The peptide. So this whole thing here, apart from this group from here like this is called a dipeptide. Now how about if I form water from here too? Like I told you that water will be formed via condensation reaction. So water will be given off H2O. So if water is given off, it means I'm removing water from here. I remove OH from here, right? and then here I remove hydrogen. So what are we going to be having? You have something like this. The water is off, right? The OH from COOH and the H from NH2 give us this water via a condensation reaction. Now what do you form here? This is another peptide linkage. Do you get it now? This is another peptide linkage. So we have two peptide linkage, and how many peptides? 3. So 2 peptide linkage is going to give us 3 peptide, which is a tripeptide. Now you are going to look at it very well. You discover that the nitrogen here, this amino group, all other amino group and carboxylic acid group are no longer present. They form condensation with each other. But look at this nitrogen here at this left hand side. You discover that amino group, you discover that it's still intact. That is why we call it the N-terminal, because it does not undergo the condensation reaction. You always see them in all protein, no matter how long the chain. There is always, at the ending, you always see this N-terminal amino group and then the C-terminal carboxylic group. Are you getting it now? So that is that. Now the smallest known protein is just 9 residue long, and that smallest protein is called oxytocin. You know what oxytocin is? It's an hormone for labor that is used for the induction of labor, which we are going to look at later. And then the largest is over 225,000 residue, the structural protein titin. So the smallest proteins you can ever see is oxytocin, 7 residue, while titin is over 25,000 residue. Titin is a structural protein. Oxytocin is a hormone that is used for the induction of what? Labor. All right, so that is that. Now, properties of protein. Let us look at some of the properties of proteins quickly. Now, the taste. Ordinarily, proteins are tasteless. However, the hydrolytic product, that is conjugated proteins, are bitter in taste. Now we'll talk about the hydrolytic product. That means when these proteins are broken down, they are bitter in taste. Are you getting it now? When you talk about hydrolytic, something is broken down by what? Water. They are bitter in taste. Ordinarily, proteins are tasteless. So this is something you can just go through. They are odorless, but when heated to dryness, they turn brown and give off the odor of a burning feather. Most times they are odorless, but when they are heated to dryness, they turn brown like sugar and then give off the odor of a burning feather. Now, the molecular weight. Proteins have large molecular weight, hence they are called macromolecules. Is that okay? Proteins have large molecular weight, hence they are called macromolecules. Is that okay? That is that. Proteins have large molecular weight, hence they are called macromolecules. Of course, except oxytocin that have 7 residue. Molecular weight of that, if you count it, if you calculate it, it may not be that much, but at least it will be higher than some other lower micromolecules. All right, so that is that. Now, the acid-base properties. Now, listen. When you were classifying amino acid in your first semester, you were taught that we have the acidic amino acids and then we have the basic amino acids. Now, you agree with me that you were told that the acidic amino acids are those amino acids that have an extra acidic group in them. For instance, glutamate and glutamic acid and aspartic acid. Those ones, they are acidic amino acids. Let me try and see. Okay, we have aspartic amino acid and glutamic amino acid. Those ones, they have the carboxyl group attached to them, apart from the normal carboxylic group. So you have something like the COOH. If this is an amino acid, let me use H. No, C. Let me use the way I wrote it before. COOH, and then here is H. Then you have C. All right, and then you have H2. another COOH. Are you getting it? This one is a carboxylic acid amino acid. So it's an acidic amino acid. Now the reason why we call it an acidic amino acid is because it has